So to start our tissue unit, I really want to kind of review cell parts and the cell life cycle and other cell processes. So starting with cell parts, or cell organelles as we call them, I want to talk a little bit about how they function and then if they play a role in a larger cellular function. So starting with the cell membrane, remember that this is a phospholipid bilayer, which means it is made up of these phospholipids here in two layers, and you can see those two layers there. And this is really to protect the cell, and it also controls osmosis and diffusion and endocytosis and exocytosis, which is the movement of material in and out of the cell. Then we have the cytoplasm. The cytoplasm really provides some structure. It also provides a solution to dissolve materials in, and because it is slightly gelatinous, it holds the organelles in place so that they don't settle to the bottom of the cell, and they stay fixed in their position so that the cell kind of knows where the organelles are going to be. Then we have vacuoles. Vacuoles are storage containers for the cell. So we can store energy molecules, or we could store cell parts that we're going to use to make larger organelles later, those kinds of things. It's just a storage place. Then we have the cytoskeleton. Along with the cytoplasm, the cytoskeleton provides structure, and this involves microtubules and actin filaments and microfilaments. Those are the three types of bones, as it were, for the cytoskeleton. And so this is really going to provide quite a bit of support and structure. They also act as material movement highways. So we have transport proteins that can walk across these fibers and kind of help move material around the cell using these as a pathway. They also are the basis for our cilia and flagellum. So if you've ever seen those hair-like projections off of a cell, those are produced using cytoskeleton as the underlying structure. And I have a little picture of that here so you can see some microvilli and how those cytoskeleton produce that. Then we have lysosomes and peroxisomes. These look very similar. What these organelles do is they break material down because they are full of enzymes. And you can see that here. So these orange dots here, those are representing enzymes. And so lysosomes and peroxisomes are both responsible for breaking things down and disposing of things, which is why lysosomes get the nickname garbage truck, because, they're transporting things around and disposing of them, as it were. And then peroxisomes are used for catabolism, which is part of metabolism, where we're breaking things down. So let's talk a little bit about energy production. So in your guided notes, if you flip past all of the organelles, you will see a chart talking about all the different cellular processes. So you'll want to make sure you add this to that. And energy production is just that. It's how we make energy for the cell. And cells use an energy molecule, specifically ATP. That stands for adenosine triphosphate. So adenosine coming from this lovely little section of RNA here. You see the ribose sugar, and then you see adenine. So we call this collectively adenosine, and then you can see the three phosphate groups. So this is where ATP gets its name. Where does that take place? Well, this takes place in the mitochondria. So the mitochondria is an organelle. It has two membranes that allow it to move electrons around in order to build ATP. And that is going to be the predominant way that we make ATP. That's the predominant way we do energy production, is through utilizing mitochondria. Now, if you'll remember, there are some other steps like glycolysis that happen in the cytoplasm and that we can carry out even if there isn't enough oxygen to utilize our mitochondria. But really, your best use of your calories is to use your mitochondria. So our next cellular process is the protein synthesis. So this is how we're going to make proteins. Synthesis meaning to make, and then protein obviously is the thing that we are making. So starting with the parts involved, this is going to start with the nucleus. This contains our genetic material, and it is going to protect our DNA and make sure it doesn't get broken, and then provide a space where the different functionality that DNA goes through can be done. But we need a little bit more than that. We need to also construct those proteins. And this is where endoplasmic reticulum comes into play. So we have two types of endoplasmic reticulum. The smooth endoplasmic reticulum is involved in making lipids, so we're not really going to talk about them right now. We're going to focus on the rough endoplasmic reticulum because proteins are the workhorses of the cell, so that's where we're going to spend most of our time. And so the rough endoplasmic reticulum is the thing that provides a space for the construction of proteins, specifically the amino acid sequences. And our next organelle, the ribosome, is going to be the machinery that actually builds that amino acid sequence. So, interesting thing about ribosomes is they are not membrane bound. They do not contain cell membrane. Like we have talked about with all the other organelles, all of those are considered membrane bound organelles, but not the ribosome. And then, like I said, its function is to actually build the sequence of amino acids. Then you have the Golgi apparatus. This is going to check our proteins and make sure that they are folded properly, that they look correct, and then it's going to send out these proteins to wherever they need to go. So it's in charge of transporting out our proteins. And the way it moves those proteins is through a transport vesicle, which is our next organelle that we're going to talk about. That transport organelle, you can see right here, is going to move around those proteins for the cell. So let's talk a little bit about how protein synthesis occurs. So starting in the nucleus here, we have RNA polymerase. It's going to unwind the DNA and then also unzip it so that we can get access to the nitrogen-containing bases in the middle. And then the RNA strand is going to be built from the template strand. Moving out into the cytoplasm here, most probably on the rough endoplasmic reticulum, because the rough endoplasmic reticulum gets its name because it is full of ribosomes. That's what gives it that rough texture, is those ribosomes. And so the ribosome is going to actually construct using the mRNA we got from transcription in the nucleus. So we have that mRNA strand here, the ribosome reading it and constructing that strand of amino acids. And that is the polypeptide chain, because amino acids are held together by peptide bonds. So we call this a polypeptide. And the interesting thing is that ribosomes don't work from one end of the mRNA to the other, and never allow any other ribosomes to work on that strand of mRNA. We have multiple ribosomes working on a single strand at any given time so that we can make lots of proteins all at once. Just a little refresher, on how to read a codon chart. So remember, you have your first letter here, and then you have your second letter here, and then your third letter here. So you're going to, whatever the codon is, you can find out the amino acid very, very quickly. And then this is possibly new information to you about protein folding. So once the ribosome creates the strand of polypeptides, so the strand of amino acids, it's not going to be folded correctly, and we need it to be folded correctly in order for it to carry out its function. So what we do is we send this to the Golgi apparatus, where we're going to make sure that everything is folded correctly. That's what the Golgi apparatus is going to do. It's going to check, and if it's not folded correctly, then it's going to be discarded and broken apart and used for other proteins later. So what are the types of folding that proteins can be involved in? So the primary folding is the initial connections between the amino acids. So that would be just like linking up the strand like this. Then after we've done that, we can get the secondary structure, which there are two types. We have alpha helixes and beta pleated sheets. So the helix, you can see there, turns into a spiral, whereas the beta pleated sheet, you can see it gets folded into an accordion style shape. And then we start stacking those up on top of each other for the tertiary level of folding. And then the quaternary folding is where we take these segments and start linking them together to make a full 3D shape that can actually carry out a function. All right, so moving on to our next cellular process. This is actually going to be two different processes. We are going to look at cell division, and we're also going to look at cell death. So remember, most cells spend all of their time in interphase, where they're carrying out normal cell functions, just making proteins and making energy. But sometimes a cell becomes sick or it becomes very, very old, and so we go through what we call apoptosis. Apoptosis is cell death, and what happens is the cell deconstructs its cytoskeleton, so it starts to implode. It collapses in on itself and forms these like cell part bubbles, and then those are going to be absorbed by white blood cells or the surrounding cells of the body. So sometimes these are absorbed by neighboring body cells. And those cell parts are then just recycled and then reused to carry out cell processes. The last cell process that we're going to talk about is cell division. And there are two types. The most common type is what we call mitosis. This is where body cells are dividing in order to replace themselves. Meiosis is a special process that's only carried out in the reproductive organs to make gametes, and we're really not going to hit on that again because that's not what is most important to us at this time. So mitosis, if you remember, is how we are going to divide the cell, and we're going to start with the organelle that's involved called the centrosome. And the centrosome is a specialized organelle that has an inner component called the centrioles, and so that's why they're sometimes referred to as either one of these. You'll see both of those interchangeably. And their main function here is to build the spindle fibers for cell division. So here you can see prophase occurring, and you can see those are our centrioles with our centrosomes in the middle, building out those spindle fibers, and those chromosomes are going to start moving towards the center of the cell so that they line up along what we call the metaphase plate, and they'll now be in the next phase of mitosis called metaphase. From metaphase, we will go to anaphase, which is where the centrioles start breaking down the spindle fibers back here towards the centrosome. So they start breaking it apart here. And that puts tension on the chromosome in order to separate it. And that's where we get separation. And then, of course, the cell membrane is going to start pinching in on itself. This is going to occur during the process of, or the stage called telophase, so that we can actually separate out the cells. And then that full separation into two cells is the process of cytokinesis. That is the separating of the cytoplasm into two different cells. And that is really the end of our cell review. All of this should have been really pretty familiar to you, except maybe the protein folding. And we are going to kind of like review in class the cell organelles to make sure that we have a little bit more familiarity with those. But really just making sure that you kind of jog your memory from what you should have gotten in biology freshman year.